I'm Ryan.
I'm Rob.
I'm Tricia.
Welcome to our YouTube channel!
Good morning, everyone. Today's experiment is entitled, What are the different membrane transport processes?
At the end of this experiment, you shall be able to investigate the different types of biological transport processes, relate osmosis, diffusion, and dialysis to the selective permeability of the cell membrane, and lastly, test the presence of proteins and sugar in the dialysis.
Indeed, it was a very productive yet long day for each one of us. Performing experiments we had never experienced before and integrating concepts we've read the night before the experiment were really a milestone for our group.
In the grand scheme of things, it is imperative to say that we have blast and so much fun.
So the following video depicts the totality on what we have done during the duration on our experiment, wherein we perform diffusion, osmosis, and dialysis. Enjoy!
These are the materials: beakers, spatula, funnel, diesel tube, test tube with rock, pipettes and aspirators, graduated cylinder, test tube brush, wire gauze, and hot plate. We also use reagents like evaporated milk, potassium permanganate crystals, and 30% sugar solution. Procedures for diffusion.
First, fill 250 ml beakers with 100 ml of water each. Second, warm the contents of one beaker. Then, drop at the same time 2 to 3 crystals of potassium permanganate into each beaker. Last, observe and compare the rate of diffusion of the solute.
Procedures for osmosis.
First, cover the large end of the thistle tube with cellophane and tie using a rubber band. Second, cup the stem of the thistle tube with finger and fill the ball of the tube about 10 cm with a 30% sugar solution. Third, suspend the thistle tube in a 500 ml beaker with distilled water. See to it that the level of sugar solution coincides with that of water outside the thistle tube. Lastly, measure with a ruler the level of sugar solution at 30-minute intervals for 2 hours and record the results.
Procedures for dialysis. First, cut 1 square foot of cellophane and soak it in water for a few minutes. Second, fold it like a filter paper to fit into a glass funnel. Third, pour 50 ml of milk in it. Fourth, collect all the edges of the cellophane and tie with string to secure them. Leave some air space above the milk. Fifth, place the bag into a 500 ml beaker and suspend it so it almost touches the bottom. Then, add distilled water to the beaker until it is at the equal level with the surface of the milk. Last, stir the water outside for 1 hour to let the milk dialyze. Let the recovered dialysate for the presence of protein and sugar.
Unfortunately, we will not able to finish the osmosis and dialysis part of the experiment due to the limited time left for us to complete the whole process. With this unexpected event, we created somehow predictions on what will be the actual results of the experiment was fulfilled.
The rate of diffusion is much greater in warm water as compared to normal one, because high temperature increases the kinetic energy in molecules.
In osmosis, the molecules of the 30% sugar solution are observed to move from a region where they are highly concentrated to a region where they have a low concentration through a semi-permeable membrane, which in this experiment, the thistle tube is covered with a cellophane in its large end.
experiment, dialysis occurs when 50 ml of evaporated milk is contained at dialyzing bag or cellophane and immersed with distilled water. When equilibrium is achieved between samples and dialysate, only small molecules dialyze out and large particles left inside the cellophane.
As a rule of thumb, in everything that we do it the best way we can. Because by doing so, we are reminded to live our life for God's greater glory.